Hello, thank you all for taking time out of your day to listen to my presentation on wetland loss in the Western Hemisphere, impacts on avian diversity known and unknown. I'm Keith Barker. I'm the Curator of Genetic Resources at the Bell Museum of Natural History and an Associate Professor in the Department of Ecology and Evolution and Behavior at the University of Minnesota. Today, I want to tell you a bit of a personal story about the interaction of collections, genetic data, and conservation, based in part on my work as well as on the work of others. In my day-to-day work, I spend a lot of time looking at things like this. Here on the left are marsh wren specimens from the Bell Museum's collections. On the right are a bunch of DNA sequences, in this case, mitochondrial DNA from rails. Looking at these things, you might think this work seems very detached from the real world. But seen in the right light, they point directly at the urgent need for conservation, concern, and action. To see things in that light, I want to walk through some projects that have focused my concern on Western Hemisphere wetlands in general, and particularly the wetlands of central Mexico. I want to start here with the work I did on the phylogeny of a diverse group of Western Hemisphere birds, the Emberizoidea. This group includes 5 traditional families of birds: the sparrows, passerellidae, blackbirds, icteridae, warblers, Parulidae, cardinals, cardinalidae, and tanagers, Thraupidae, as well as a number of relict groups, including the yellow-breasted chat, wren thrush, and others. In this project, in collaboration with colleagues at Cornell, San Diego State University, and UNLV, we were seeking to create a species-level phylogeny for about 804 species, one of the largest such trees to date. We tackled this project by a combination of higher level studies, including representatives of all these groups, and by doing individual studies splitting up the families. I took primarily responsibility, along with Scott Lanyon, for the phylogeny of the icteridae, the blackbirds and relatives. There has thankfully been little recent extinction in this group. As you can imagine, studying the phylogeny of an extinct species using molecular data poses some challenges. Unfortunately, the icteridae is one family where extinction has occurred in historic times. Specifically, I'm referring to this species, the slender-billed grackle. This is a form that was described by Swainson in 1827, based on specimens collected by William Bullock in the Valley of Mexico, basically the valley that's now filled by Mexico City. This bird is clearly a close relative of the great-tailed grackle, which now occurs here in Minnesota, but historically was restricted to central Mexico, and the boat-tailed grackle of the Gulf and East Coast in the US. It's distinguished by coloration of the female plumage, as well as body and especially bill size and proportions. The species was later found in the adjacent high mountain valley of the Rio Larma, near the city of Toluca, just west of Mexico City, and was last seen and collected there in 1910. It is now presumed extinct. As part of our work on blackbird phylogeny, we sequenced mitochondrial DNA inherited through the maternal line in birds, from an early 20th century specimen, and analyzed it along with DNA from other grackles. This work was led by Scott's graduate student Alexis Powell and published in 2008. What we found was kind of surprising. The slender-billed grackle, up here highlighted in red, it was related to some, but not all, great-tailed grackles. There was 2 major genetic groups or clades of great-tailed grackle DNA. Those are shown here in this gray shading. One close to the slender-billed grackle, and the other was more closely related to the boat-tailed grackle. Here's what this situation looks like in cartoon form. Interestingly, there's evidence from a hybrid zone that boat-tailed grackles and great-tailed grackles are what we call biological species. They have reduced hybrid fitness and prefer to assortatively mate. As far as we know now, the eastern and western clades of great-tailed grackles freely interbreed with one another in the southwest of North America. We don't know for sure whether the slender-billed and great-tailed grackles ever interbred, but it seems at least possible, given that even great-tailed and common grackles have interbred occasionally in the past. It's likely that the eastern and western clades of great-tailed grackles were once isolated lineages that retain the ability to interbreed, and now they're fusing since they have come into secondary contact. Eventually, one or the other of these two lineages will go extinct, and the great-tailed grackle will look genetically like the sister species of either the boat-tailed grackle or the slender-billed grackle. What has this got to do with conservation? Well, obviously, the slender-billed grackle is extinct. And it lived, as far as we can tell, only in two places in the world. One of them was here in the Valley of Mexico. This was the heart of the Aztec Empire, with its seat at Tenochtitlan right here. The valley was filled with rich wetlands, with very fresh water here in Chalco, in Xochimilco, and here in Lago Texcoco to the west of the dike of Netzahualcoyotl, which was emplaced there actually to separate the fresh water from the more saline waters of Lake Texcoco, Saltocan, and Sympongo to the north. From later observations in the Rio Larma Valley, the slender-billed grackle specialized in rush-dominated freshwater marshes, which would have dominated Chalco, Xochimilco, and western Texcoco, also known as Mexico. By the mid-19th century, these wetlands had been extensively reduced by drainage for agriculture, deforestation, and erosion, infilling of the lake basins, and possibly by increasing aridity due to, at that time, non-anthropogenic climate change. You can see that Mexico is gone, the Lago de Mexico. Texcoco is no longer connected to Xochimilco in the south. Here's Xochimilco and Chalco. Or to Saltocan or Sympongo to the north. Slender-billed grackles likely persisted in Xochimilco and Chalco at this time in the mid-19th century. In modern times, nearly the entire wetland complex is covered by the Ciudad de Mexico, Mexico City. On the left here is a map of the modern city with ancient lake beds superimposed over the map. And on the right is a satellite image of approximately the same area. Almost none of these original rich wetlands still exist. In the north, a small remnant of the Lago Sympongo exists. This is a highly polluted water with industrial and domestic waste and dominated by carp. To the east, you can see the end of Mexico City's Grand Canal, which is essentially an open sewer that disappears into the ground here in the northeast corner of Sympongo to be piped north to the river basin, where it is partially processed and then released into the river. There is no wetland habitat on the margins of this lake. Here to the east of the city is the remnant of Lago Texcoco. Most of the lake bed has been taken by urban development. Only areas that flood intermittently are left unoccupied. The remaining water is saline and polluted, and very little viable marshland habitat remains here. To the south, Lake Chalco is nearly desiccated, and much of its lake bed has been taken by urban development. As with Texcoco, there's almost no remaining marsh habitat. The only viable marsh habitat in the entire complex is here in the remnants of Lago Xochimilco. Specifically, the Laguna de Plawak is the remnant of eastern Xochimilco. Many marsh birds that have historic records from Chalco and Texcoco persist here at Plawak, and this is probably the best marsh habitat left in the entire valley. eBird, the online community science database maintained by the Cornell Lab of Ornithology, reports 223 species from this marsh, including grebes, rails, red-winged blackbirds, yellowthroats, and other marsh birds. This site has received designation under UNESCO's Ramsar Convention, the Convention on Wetlands of International Importance, but is not a protected area. Clearly, since the Spanish conquest, urban development in the Valley of Mexico has massively impacted the wetlands of this area. In fact, the effects don't end in the valley. As I noted earlier, the effluent of the city is actively pumped out of the basin, since the basin is slowly sinking as water is drawn from the aquifer to supply the city. Also, a significant percentage of the city's water supply is imported from adjacent basins, including the headwaters of the Rio Larma in the state of Mexico. The Valley of Mexico is one of several endorheic basins in central Mexico. These are basins that have no outflow to the ocean, eventual outflow to the oceans. And these range from the Magdalena Basin in the far west here to the Llanos de San Juan in the east. These basins are continually formed by volcanic activity in this region, which is referred to as the transvolcanic belt. Lava flows, so here's the transvolcanic belt here. Lava flows from these volcanoes, indicated on this map here as triangles, block water flow, forming lakes or marsh complexes until erosion or tectonic activity eventually opens them to drainage once more. Adjacent to the Valley of Mexico, shown here, is the headwaters of the Rio Larma. You can see here, it's labeled, comes right up here just west of the Valley of Mexico. This is the other locality from which slender-billed grackles were known prior to their extinction. The Rio Larma flows west into Lago Chapala, from which it emerges as the Rio Grande de Santiago, and then flows out into the Pacific. It's likely that agricultural and other land use changes in the Larma River Valley contributed to the elimination of the slender-billed grackle sometime after 1910, the last time it was collected. Given the sparse collecting in the area until the 1960s, it could conceivably have happened any time between 1910 and 1960, a brief period of 50 years. In particular, changes in the Rio Larma Valley likely accelerated in the 1940s, so the species may have persisted until then. The Systema Larma is an aqueduct system begun in 1942 and expanded in 1967 that pipes water from about 300 wells in the Larma Valley and pumps that water over the mountains into Mexico City. So here's Mexico City. You see in dark blue is the Systema Larma, which has its origin in wells in the Larma River Valley, and that water is pumped over the mountains into Mexico City. More recently, another system called the Systema Cutzamala pipes water from further west and provides much more water than the Larma. And here's the Systema Cutzamala, which again pumps water into Mexico City. Continued extraction from the Larma has pulled the water table down to where wells now need to be drilled down to 40 meters, whereas within living memory, the water table is nearly at the soil surface. Continued drawdown in the Larma is putting these marshes at risk. Even so, it's not enough for, that is the water supply to Mexico City. During dry periods, water service is regularly interrupted in Mexico City, and almost all homes have cisterns to store water that is delivered by truck. Hundreds of millions of pesos are spent on pumping water, and it takes as much power as the entire city of Toluca, a city of over 2 million people to the west of Mexico City, to keep the pumps going. This situation is extremely volatile, and it is far from certain what the future holds for this region, despite the few protections that are in place. Once a massive wetland complex, similar in scale to that in the Valley of Mexico, it nurtured multiple pre-Hispanic cultures at its margins. The wetlands here have been massively reduced by urbanization and over-extraction of water. Originally, 3 large lake complexes filled these basins to the east of Toluca. And here I've outlined these in red, including Chiconahuapan in the north, Chimaliapan, or also known as Lerma here in the center, and Chignahuapan in the south. Chiconahuapan has been nearly completely drained, and only a small but very rich marsh remains here. The largest marsh remnant is here in the center of the valley. The southern portions of the lake have been extensively drained and converted for agriculture. And the former main source of water for the marsh, the Rio Larma, can be seen to the left here in its canal. To the south, there's also a small remnant of Chignahuapan, which also remains rich in species diversity. These three wetlands are part of a reserve complex called the Área de Protección de Flora y Fauna Cienegas del Lerma. This site has received designation under the Ramsar Convention, as I mentioned earlier, and is considered an area of importance for conservation of the birds of Mexico. However, all these wetlands are under significant threat, not only due to agricultural conversion and pollution, but also because the groundwater of this region provides 5 to 9% of the water used by Mexico City. Well, we've come a long way from the slender-billed grackle and its two known localities shown here in the Valley of Mexico and the Rio Larma Valley. Although the plight of these marshes is dire, the species is already extinct. Does the avifauna of this region, and especially its evolutionary history, give us other reasons to worry about the future of these sites? The answer is, as you might imagine, yes. The next encounter I had with the importance of central Mexican wetlands was through my work on the phylogeny of wrens. As part of an ongoing effort to sort out the phylogeny of all wren species, I received funding from NSF not only to work on higher level relationships, as shown here in this paper from 2017, but also to explore genetic variation within some wren species. One of the species we chose to work on is the marsh wren. The marsh wren is a widely distributed North American species found in cattail and rush marshes. It's ubiquitous in Minnesota. From northern Canada down through central Mexico. You can see this little region of purple indicating year-round residency in central Mexico. It's a highly polymorphic species with 14 described subspecies, suggesting that its current taxonomy might not match its evolutionary history. And so we've been studying genetic variation across the range of this species. We've sampled DNA sequences from, as I said, from across its range, including non-migratory populations on the West Coast, the Gulf Coast, and here in central Mexico. I'm showing here the localities from which we currently have data. This includes the first ever samples from Mexico, which we collected on an expedition in 2012, plus a wide sampling across the species range, especially in the east. We're in the process of adding more samples to this study, including material we collected from Montana and Nevada. Here's a tree of our mitochondrial data so far. You don't need to see the labels to understand the basic structure. What we see here are two deeply divergent lineages with three major lineages within this lower lineage here. These two lineages are about 8% divergent, which is approximately the same divergence as between humans and chimpanzees at this particular locus. These two lineages are definitely distinct species that can be diagnosed using song cues that you can hear with your own ears as long as you know what you're looking for. The Western marsh wren lineage is divided into three main lineages, about 2% divergent from one another, which is a pretty typical divergence for species level differentiation in North America. This includes an interior lineage represented here by sequences from Nebraska and interior Washington, a coastal lineage down here from California birds in coastal Washington, and then this transvolcanic lineage, which from central Mexico. This lineage includes all the birds that we collected in 2012. It looks like we have strong data supporting at least two species of marsh wren, eastern and western, and I would argue for 4 different species, recognizing each of these western lineages. We know, for instance, that there are significant song use differences between transvolcanic and western interior wrens, and there are probably differences between western and coastal interior birds. And actually have a student, Isaiah Clark, who's been working on this question, and we do indeed see differences between western interior and western coastal birds, in song specifically, but not something easily discerned in the field. Taxonomically, here's what things will look like if we recognize the 4 marsh wren species. The eastern birds will be palustrous, the western interior birds Plesios, the western coastal birds Paluticola, and the central Mexican birds Toluquensis, or the Toluca wren. Of course, it will be almost impossible to recognize these birds in the field, except for distinguishing members of eastern and western clades by song. As I mentioned, we now know that we can distinguish western interior and coastal forms using song data, but this can't be done by ear. However, Telekinesis, the endemic Mexican form, is quite distinctive in its underbody coloration. That's shown here, where we have 2 Plesios and 2 Telekinesis individuals from the same central Mexican marshes. The most distinctive character is the underbelly and chrism coloration, shown here, which are much darker in Telekinesis than in Plesios. You can see there's also some differences in back color for these individuals. I noted that these birds, oh, actually, I didn't note that. I should have noted that these birds were from the same marshes, actually, collected. These are birds we collected in 2012. These are birds from some of the same marshes. Western Plesios actually winter in central Mexico. So you're only guaranteed to see the Toluca wren if you're birding in central Mexico from June through August. Here's the Bell Museum's collections of marsh wrens from Mexico. This drawer right here. I've sorted the specimens out by month. And we have specimens actually from all months except for February and December. You can see the birds from May through August, here through here, have very dark underparts, whereas birds collected in October, say, and November are a mix of light birds and dark birds, representing wintering western birds and the local endemic Toluca wren. We've genotyped every single bird in our collection using a simple genetic assay, and they can easily be split into wintering and resident birds. So here's wintering Plesios, and here's resident Toluca wrens. Turns out that we have the world's largest collection of Toluca wrens right here, due to collecting done by Bob Dickerman and Juan Nava in the 60s and 70s. The only other collections with any of this species are at the Cornell Museum of Vertebrates, the Delaware Museum of Natural History, and the Smithsonian, and now the Zoology Museum at UNAM in Mexico City, where we deposited half of our specimens from the 2012 expedition. If we plot the specimens of this endemic form, we get this distribution. These little green dots show specimens. The Toluca wren is endemic to the eastern portion of the transvolcanic belt, as far north as Hidalgo, as far south as the southern Rio Larma Valley, as far east as the Llanos de San Juan, and as far west as the middle reaches of the Rio Larma. Notably, this species occurs at the 2 sites from which we know the slender-billed grackle, the Valley of Mexico and the Rio Larma Valley. Based on specimen data, this species appears to be absent from extensive wetlands further west in the transvolcanic belt. Looking at summer eBird records of marsh wrens from central Mexico, when most, if not all, wintering birds should be gone, we do find some evidence of marsh wrens further west, specifically Lake Quetzal, the Laguna de Zacapu, a very small wetland remnant of what was once a very large freshwater lake, and the Lago Pátzcuaro. Unfortunately, we don't have specimens or genetic data from here to confirm that these are actually Toluca wrens, or whether they might be late lingering migrants from the north. It would be great to confirm these records. I mentioned that this species is present at the sites from which the slender-billed grackle was extirpated. Looking at observational data compared with specimen data, we also see some sites where wrens appear to have disappeared. In particular, the endorheic lakes of the Llanos de San Juan, called Tottosingo and Tepeahualco, no longer have wrens. This becomes clear when you look at satellite imagery, or why that is becomes clear when you look at satellite imagery of these areas. This is a recent satellite image of the region showing both lakes. These basins were heavily converted to agriculture through the 20th century, and the groundwater that supported them was tapped extensively by Mexico City starting in the 1970s. Both lakes, Tepeahualco here and Tottosingo here, have been totally dry since 1993. There are still some limited wetlands southwest of Tottosingo, which you can see sort of here. But it's unclear how diverse they are and what species occur there. Specimen data clearly show that Toluca wrens once occurred in Tottosingo, as did a diverse fauna of ducks, rails, grebes, herons, and other water birds. Almost all of these species are now extirpated. Similar extirpations could easily happen in the handful of other Toluca wren localities. One such site is Lake Texcoco in Hidalgo state, northeast of Mexico City. This extensive marsh complex has fluctuated widely through the 20th century due to changes in rainfall. However, deforestation and agricultural modification of surrounding lands has increased sediment deposition over the long term, leading to infilling of the lake. Dikes, as you can see here extensively, have further fragmented the lake and lead to more rapid evaporation and salination. Although this site has been designated under Ramsar, it is not protected and is changing rapidly. So it's not just the slender-billed grackle and the Toluca wren that are endemic to these threatened wetlands. Here's a phylogeny of the genus Geothlypis, which includes the yellowthroats, mourning, Miculabraes, and Kentucky warblers. This is not my work, but that of a colleague from our Emberizoid work, John Klicka, and his colleagues, including Patricia Escalante at UNAM. One of these species is the black-polled yellowthroat, Geothlypis speciosa, shown here, which is the sister group to almost all other yellowthroats. This species is classified as globally endangered by the IUCN. The species is endemic to some of the same regions as Toluca wren, including the Valley of Mexico and the upper Rio Larma Valley, although it appears to have been extirpated within historic times from the Valley of Mexico. We have specimens of this species from much further west than the Toluca wren, including these wetland areas here, more similar to recent eBird observations of the Toluca wren. We have similar distribution for eBird observations of this endangered species, although we see some problem misidentifications up here in Guanajuato and over here in Teotihuacan, where there's no wetlands. So it's not too likely that a species occurs there. We also see from observational data that the species appears to be absent from the Valley of Mexico, where specimens tell us it once did occur at the margins of Lago Texcoco. There are a number of other avian forms endemic to the wetlands of this region, as well as some mysteries. I want to mention one last intriguing case, again from the Rio Larma Valley. So right here. The yellow rail is a species I hope you're all familiar with from here in Minnesota. As shown on this range map, the species breeds in north central North America and winters primarily on the Gulf and Atlantic coasts. You can also see a small breeding population here in southern Oregon and northern California that winters on the California coast. What's not shown on this map is a possibly extirpated breeding population of this species in central Mexico, marked here with this red star. These birds occur in the upper Rio Larma Valley, in the same marshes that used to have slender-billed grackle and still have Toluca wrens and black-polled warblers. Evidence for this population comes from a handful of specimens in a few collections, including these 5 from the Bell Museum, which includes a series of 2 adults and 3 juveniles. Based on a single specimen collected by Goldman that's deposited in the Smithsonian, Nelson described this population as a new species, Guternakops, at the time Porzana, goldmani, named after the collector Goldman, in 1904. Most recent specimens of this population were collected by Juan Nava in 1964, deposited here at the Bell as well as at the Delaware Museum of Natural History, Moore Lab of Zoology in California, and LSU. And one specimen was collected in 1970 that's deposited at Cornell University. eBird has no records of the species in Mexico, and it is likely that this population is extirpated, given the extensive modification of the wet grassland habitats the species prefers. There's currently no genetic data for this population, although a broad survey of genetic variation across the species' northern range showed very little differentiation from Oregon through Quebec. It's entirely possible that this is yet another deeply divergent endemic Mexican form. We'll just have to see. So I want to conclude by thinking more broadly about wetland conservation in the Western Hemisphere. The most recent estimate of global inland and coastal wetland area is in excess of 12.1 million square kilometers, an area larger than the country of Canada. The Western Hemisphere harbors about 43% of these wetlands, ranging from the Mackenzie River Delta to the plateau lakes of Patagonia. As I've shown in this talk, the wetlands of central Mexico have preserved an array of deeply divergent endemic bird species, which is true of other such wetlands, especially in the tropics. The upland plateaus of Patagonia produced a new and spectacular, I must say, species of grebe as recently as 1974. This species is critically endangered because of climate change that are altering the distribution of upland wetlands, as well as introduced mink, which escaped from captivity in Patagonia and are extremely important nest predators for the species. The tangled marshlands of coastal Brazil yielded a new species of ant wren as recently as 1995. The fact that both of these species are endangered says something about the state of our wetlands habitats. The World Wildlife Fund's Living Planet Index is a measure of the state of the world's biological diversity based on population trends of vertebrate species. Over the last 40 years, this index has been declining much more rapidly for freshwater species than for either marine or terrestrial species. Up to 87% of the global wetland resource has been lost since 1700 in places where data exist. This may not reflect the actual global total, with rates of loss increasing in the late 20th century. Nearly 70% of wetlands present in 1900 have been lost, and they are still being destroyed 3 times faster than forest habitats. Rates of loss are particularly high in Latin America and the Caribbean, where much of the endemic wetland species diversity of birds occurs. Lest you think I'm unfairly singling out Latin America, similar and even more extreme changes have happened in the US. Here's a reimagined pre-colonial image of California, showing the largest wetland complex in the Western Hemisphere, Tulare Lake. Due to drainage and conversion to agriculture, in part motivated by the desire to suppress native peoples of the Central Valley, this lake totally disappeared by 1898. No endemic forms are known from these wetlands, but it's entirely possible they existed. Museum specimen, or to conclude, I'm going to make a couple of points. Museum specimens document many isolated populations of wetland birds. Absence of these birds in sites where specimens show they occurred reveal many cases of extirpation, in some cases extinction. These isolated populations are often deeply divergent, representing endemic species. These wetlands, like wetlands worldwide, are profoundly threatened by anthropogenic change. And to end, I just want to acknowledge work of collaborators, including Alexis Powell and Scott Lanyon, with whom I worked on blackbird phylogeny, and ongoing work on marsh wrens that's been supported by Mark Robbins at KU, Hernan Vazquez Miranda, my former PhD student, who's now at the Instituto de Biología at UNAM, and volunteers and employees in the lab, including Joe Lutz, Isaiah Clark, and Elena Friedrich. And finally, support from the University of Minnesota and from some NSF grants that have supported parts of this work. And with that, I'll thank you all again for sharing some time with me, and I'll look forward to taking your questions next Saturday. Thanks.